Hello everyone, today we are going to talk about membrane transport. A solution is made out of two parts, a solute and a solvent. The most common solvent we use in lab is water. Solutes, we are going to talk about three types of solutes: colloid, crystalloid, and suspension particles. The smallest particles out of those three would be the crystalloid, then comes the colloid, and then the largest would be the suspension particles. An example of a crystalloid would be iodine. If you add iodine to water, you are going to be able to see through it because those iodine particles are really small. They're going to dissolve in the water. So when you're able to see through it, that would tell you that the particles in that solution are crystalloids. If you can't really see through it, so it's pretty opaque and so the light is not shining through it, that would tell you that these particles are a bit larger and they are colloids. For example, if you are looking at a test tube filled with milk. Suspension particles, they tend to settle out and precipitate. So when you have precipitate, that would tell you that those are suspension particles. There is a concept known as the Brownian motion. What is the Brownian motion? So the Brownian motion is basically explaining to you that particles, they have kinetic energy. And that kinetic energy causes a rapid random movement of those particles. You can see right here, the particles, they start to bombard against each other, and that causes the particles, especially if we are looking at a solution, that's what keeps the particles suspended in the solution. One more thing to mention about the Brownian motion is that when you actually look at the particles under the microscope, you are actually going to see the particles vibrating, and that's actually the Brownian motion. Diffusion is a passive transport, meaning that it does not require any energy. Diffusion is basically the movement of the solutes from a high concentration area to a low concentration area. For example, if we have side A and side B, and side A, I went ahead on this side, I went ahead and I sprayed perfume. So here are the perfume molecules. Now notice how side A has a high concentration of the perfume molecules. What's going to happen is that those perfume molecules are going to start diffusing over to side B, which has the lower concentration of the perfume molecules. And this movement is going to continue taking place until both sides are equal. So until you have equal concentration of the perfume on both sides. So this movement, it's passive, it does not require any energy. Now there are some factors that can actually affect the diffusion rate, either increase it or decrease it. For example, temperature. The higher the temperature, the faster the molecules are going to diffuse. So the higher the temperature, the higher the diffusion rate. That is a directly proportional relationship. Another thing is the particle size. The larger the particle size, the slower it's going to move. And so therefore molecular weight or the particle size has an inverse relationship with the diffusion rate, because as the particle size increases, the diffusion rate is going to decrease. Another thing actually to mention is that the greater the concentration gradient between two sides, the faster the diffusion rate is going to be. Meaning if side A and side B, again, there is a huge difference, in terms of how many perfume molecules are on side A or on side B. The greater the difference between the two sides, the faster the molecules are going to diffuse to basically reach equilibrium, because the particles would want to reach equilibrium so that both sides would be equal. Right here we have an agar plate where part of the agar was cut out and a drop of dye was placed in each hole. We leave the plate for an hour or so, and we keep observing it to see what's going to happen with the diffusion of the dye molecules. Now notice how this dye right here is diffusing faster compared to this one. This one is potassium permanganate, and this dye right here is methylene blue. Notice how methylene blue is not diffusing as fast, and the reason why methylene blue is not diffusing as fast as potassium permanganate, because methylene blue is actually larger. So its molecular weight is 320 compared to potassium permanganate, which is smaller and lighter. Its molecular weight is 158. So one more time, this kind of shows that the heavier the molecule is, or the larger it is, the slower it's going to diffuse. And again, that's an inverse proportional relationship between the diffusion rate and molecular weight. Dialysis is a passive transport, so no energy is required. If energy is required, by the way, we call that an active transport. And the reason why active transport would need energy, because the solutes are going to be moving from a low concentration area to a high concentration area. So that would be an active transport. Dialysis is passive, so no energy is required, and the solutes are going to move from a high concentration area to a low concentration area, which you can refer to it as down the concentration gradient. So if you ever see the term down the concentration gradient, that just means the molecules are moving from high to low. Dialysis, those solutes, they are passing through a semi-permeable membrane. That's really what makes the big difference between dialysis and diffusion, because everything else is exactly the same between dialysis and diffusion. They're both passive. The solutes in both cases are moving from high to low. The only difference is the semi-permeable membrane. So for example, the perfume example where the perfume molecules are just diffusing in the air, so we don't have any semi-permeable membranes in the air, that would be known as diffusion. But if molecules are, let's say, passing through a semi-permeable membrane, kind of like the plasma membrane of your cells, that would be known as dialysis. So this is an example right here that we usually do in lab, where we get a beaker, and in this beaker we go ahead and we add some water and we add some Lugol. Lugol is basically iodine, and that's why the water kind of turned yellowish in color, because we've added the iodine in the water. And then we get a dialysis bag. So it's a bag that has pores within it, and we fill that bag with glucose and we fill it with starch. Now you could fill it with other things, but this is just an example. So this bag right here is filled with glucose and starch. And then we go ahead and we leave it for about an hour, and we keep observing it to see what's going to happen. So notice right here how the bag is turning black. The solution outside is not changing color, but the bag continues to turn darker in color, and eventually the whole bag is going to turn black in color. Now, what does that tell you? It tells you that something is diffusing into the bag, causing that color change. Well, what do we have in the bag? We have glucose and we've got starch, and outside we have water and iodine. The thing is, um, iodine, when it combines with starch, so when iodine reacts, I should use the term reacts. So when iodine reacts with starch, starch is going to turn black, like a very dark black color. So right here, since the starch inside of the bag is turning black, what does that tell us? It tells us that iodine was able to pass through the dialysis bag and basically enter the dialysis bag and react with the starch, turning the starch black. Now let's take a look at the color outside. The color outside remained yellow. So we have iodine outside. So what does that tell you about the starch? Did the starch leave the bag? The starch did not leave the bag, because if starch had left the bag, and you have iodine outside, then outside the bag should also turn black. But because it didn't turn black, we know for sure that the starch did not leave the bag. Now, let's think about why was iodine able to pass through the dialysis bag and starch was not able to pass through the dialysis bag. The thing is, it has to do with the size of those particles. The smaller the particle size, it's going to be able to pass through, because that dialysis bag has very tiny pores within it. So only small solutes are going to be able to pass either in or out of the bag. Large particles are not going to be able to pass through. So since starch was not able to pass through, that tells us that starch is actually a pretty large molecule, and we refer to it as a macromolecule. And that's why it was not able to pass through. And so this test right here for us to figure out whether iodine passed in or out, and same thing for starch, we call it the iodine or the Lugol's test. So iodine is yellowish, kind of brownish in color. So if iodine does not react with starch, iodine is going to remain kind of brownish in color, and that would tell you that there was no starch present in that test tube. Let's say we have a test tube and it has some starch, and you go ahead and you add a couple of drops of iodine. If there is starch, it is going to turn black in color. And again, right here, iodine, when it reacted with starch, it did turn black, and that was what allowed us to figure out that iodine did enter into the bag. Now, we have glucose inside of the bag. We want to figure out whether glucose was able to pass through and basically exit the bag, and that would tell us that glucose is small enough to pass through the pores. And to figure that out, we go ahead and we perform a test known as the Benedict test. So the Benedict test and the Lugol test, those are biochemical tests that we can do to figure out the movement of solutes. So the Benedict test is a biochemical test that's going to help you figure out the presence of glucose. So what we do is we go ahead and we take a sample of the solution outside of the bag, and we go ahead and we perform the Benedict test. Now, the Benedict is blue in color. So when you add Benedict, let's say you have a test tube, and let's say I go ahead and add just water in that test tube, and then I add a few drops of Benedict to this test tube containing water. It is going to turn blue because that's the Benedict chemical color. It's blue in color. If it remains blue, that tells you that there is no glucose in that test tube. Let's say I get 3 more test tubes. In this test tube, I go ahead and I put, let's say, a drop of glucose. Over here I add like 5 drops. Over here I add 10 drops of glucose. And then I go ahead and I add a drop of Benedict to each test tube. If you get any of these colors, green, yellow, orange, and red, that tells you that there is glucose present. If it turns green, it tells you that you have a bit of glucose. If it turns orange, it tells you that you actually have more. If it turns red, that tells you that you have a whole lot of glucose. And by the way, the Benedict test doesn't just test for glucose, it tests for any monosaccharides. So for example, glucose, fructose, galactose, all of these monosaccharides, you can go ahead and perform the Benedict test if you would like to figure out if you have any of those monosaccharides. So right here, another way, instead of putting the solution in test tubes, we sometimes have those glucose test strips, which are way easier, way faster. So all you do is just take that test strip and you basically dip it in the solution, and then you go ahead and you take a look at the color that you get. So right here, you notice that after the glucose test strip was submerged in the test tube, it did change color, and you got that kind of greenish color, which tells you that you do have glucose. So that's one way of testing it. Just dip the glucose test strip, or basically take some of the solution right here outside of the bag and add the Benedict solution, and if you get any of those colors, that would tell you that you have glucose outside of the dialysis bag, which would tell you that glucose was able to pass through the dialysis bag, and that would tell you that glucose is not a very large molecule. That's why it was able to pass through the dialysis bag. Now, if we want to figure out whether water is going to enter into the bag or basically exit the bag, and the water movement is known as osmosis. So osmosis is the movement of water. All we would do is basically weigh the bag. So we don't have a biochemical test to test for osmosis. And so all we could do is just weigh the bag. If the bag weighs heavier at the end of the experiment, that would tell you that water entered the bag. If the bag basically weighs, it weighs less, it's lighter, that would tell you that the water exited the bag. So you would just weigh the bag, and that would tell you what happened with the movement, of water. And again, the movement of water is known as osmosis. Tonicity is basically, we use that term when we are comparing the concentration of two solutions. And we are basically comparing the concentration of the solutes inside of the cell versus outside of the cell. We use 3 terms, isotonic, iso meaning the same. So isotonic meaning that the amount of solutes, or the concentration of solutes outside of the cell, is equal to the concentration of solutes inside of the cell. Hypotonic is the term we use to explain that outside of the cell, we actually have a lower concentration of solutes compared to inside of the cell. So when we are using tonicity and we are using those terms, we are always trying to explain the condition outside of the cell. So one more time, if the condition outside of the cell is exactly equal, to the condition inside of the cell, we call that isotonic. If the condition outside of the cell has a lower concentration of solutes compared to inside of the cell, we call that hypotonic, hypo meaning low. If the condition outside of the cell has a higher concentration of solutes compared to the inside of the cell, we call it hypertonic. And hyper meaning high. So hypertonic meaning you have high solutes outside. Hypotonic meaning you have low solute concentration outside of the cell. Now, with osmosis, as we said, osmosis is the movement of water. And water always likes to move basically from the low solute concentration to the high solute concentration. Water likes to dilute things, so it's always going to want to go to the side that has a higher solute concentration so it can dilute it out. Now, if we are talking about the water movement, in an isotonic solution, so let's say here's a cell and you placed your cell in an isotonic solution. Again, isotonic meaning that the concentration outside is exactly equal to the concentration of the solutes inside. That would tell you that the water in this case is going to move in and out at an equal rate. And therefore, if you look at the cell under the microscope, you shouldn't see any size change. Now, if a cell was placed in a hypotonic solution, so hypotonic meaning that outside of the cell, you have a higher water concentration and you have a lower solute concentration. Water likes to go from high water to low water, or basically from low solute to high solute. So since you have the low solute outside of the cell, and inside of the cell you actually have a higher solute concentration, the water is going to start entering the cell from the low solute concentration to the high solute concentration. If you look at the cell under the microscope, you will notice that the cell is going to start swelling up because all of that water is rushing into the cell. Hypertonic solution, the water is actually going to exit the cell. And the reason why the water is going to start exiting the cell, because again, water likes to go from low solute concentration to high solute concentration. And as you can see, definitely outside the cell over here in this example, you have the higher solute concentration. So water always likes to go to the higher solute concentration side. And so the water is going to start moving out, and therefore the cell is going to start shriveling up. So to recap, in an isotonic solution, basically the cell is going to stay the same size because the water is moving at an equal rate. With a hypotonic solution, as we said, the cell is going to gain a lot of water because all of that water is rushing into the cell, so the cell is actually going to start swelling up. In a hypertonic, if a cell was placed in a hypertonic solution, the cell is going to lose a lot of water and the cell is actually going to start shriveling up and shrinking. We can see the effects of osmosis on our, on human cells. For example, if you prick yourself and put some of your red blood cells on a slide and then put some isotonic solution on some of your red blood cells and then look at it under the microscope, the red blood cells are going to look absolutely normal. But if you go ahead and you place a few drops of just pure water on your red blood cells, and that would be a hypotonic solution because your cells do have some solutes. So if you just submerge your cells in just pure water, zero solutes, all of that water is going to start entering into your red blood cell, and under the microscope, it's actually going to be swollen, and to the point that it could actually burst if it took too much water. If you go ahead and put a few drops of a hypertonic solution on top of your red blood cells and look at it under the microscope, you will notice that your red blood cells are all shriveled up because they're going to lose a whole lot of water. So if we are thinking of human cells, which are basically animal cells, the best condition for the cells would be to be placed in an isotonic solution. For plant cells, it's actually the opposite, or not the opposite, it's just a bit different. For plant cells, they love to be placed in a hypotonic solution. So they actually like to take a bit more water in. They're not going to burst because they have a pretty strong, sturdy cell wall that animal cells don't. So animal cells, we don't have a cell wall. Plant cells do have that cell wall right there. If a plant cell was actually placed in a hypertonic solution, it is going to lose a lot of water, and we call that process plasmolysis. This is an example we usually do in lab where we get 3 eggs. One is placed in an isotonic solution, so you're going to notice that size-wise it's not going to change. This one, notice how the egg swelled up. So what does that mean? It means that this egg was placed in a hypotonic solution. That's why it's swelling up. Notice how here this egg is actually shriveling. And the reason why this egg is shriveling, because that would tell you this egg was losing water. So that means this egg was placed in a hypertonic solution. Filtration is a process that's used to remove particles from a solution. So it allows liquid to pass through. It allows maybe very tiny, small particles to be able to pass through because that filter membrane does have small holes within it. So it allows, again, anything small or liquid to be able to pass through. Now, those particles that are going to be able to pass through, they are moving from a high hydrostatic pressure. Hydrostatic basically means fluid. So they're moving from a high fluid pressure to the low hydrostatic pressure. So for example, if you have a beaker right here filled with some water and solid particles, and you go ahead and you pour it down that filter membrane, which looks something like this, the filter membrane is going to hold back the larger particles and they're not going to be able to pass through. And again, with filtration, the particles are moving from the higher hydrostatic pressure to the lower hydrostatic pressure. Where is an example of where filtration takes place within our body? For example, within our kidneys. So we know that our kidneys filter out our blood, allowing the particles that you don't want to keep in your body. So like the waste, they allow the waste particles to actually exit the body and basically exit your kidneys. And then anything that's pretty important, like your cells, etc., large nutrients, all of these are not going to be able to pass through the tubules of the kidneys, and basically they're going to be pushed back into our bloodstream. And again, the reason why this is happening within our kidneys is because the pressure in our capillaries, the pressure in your blood vessels, like those capillaries, it's actually higher compared to the pressure in your kidney tubules. So again, that's going to allow the fluid, like, you know, and the little particles. So in terms of your kidney, it's going to allow the urine to basically pass through. And then again, all of the good stuff like glucose, and then of course all of your cells. We don't want to be losing our cells every time we void our urine. All of this is going to return back to the bloodstream.